This is DNA. DNA stands for deoxyribonucleic acid and has been proclaimed as the language of genetics. It is made of 4 types of nucleotides, as shown. Chromosomes play an important role in the living cell, as it allows DNA to be replicated and passed on to offspring. However, DNA can also be changed by things called mutations, some of them which are good, and some of them which cause death. For example, on chromosome 4 in a human, a gene usually contains a certain pattern that is repeated over and over again. However, if this gene is missing, the human will suffer a premature death. This gene can also be repeated too much, which causes dementia at certain ages. The reasoning behind this is because the mechanism that normally regulates the replication can get confused, and will replicate extra amounts of the gene since it is the same sequence over and again. As you can see by these drawings, Huntington's disease is purely based on how many times the pattern CAG is repeated on chromosome 4. And at 50 times, neural function decreases dramatically at age 27, no matter what external factors influence life. This of course brings up the ethics of telling patients what their DNA says. Should doctors tell their patients based on their genetic code how long the patients are going to live, or diagnose them based on genetics? This is the X and Y chromosome. The X chromosome is the 8th largest chromosome in the body and is either paired up with the Y chromosome or another inactive X chromosome. They are known as the sex chromosomes because they determine the sex of an individual. The X chromosome specifically has the gene DAX, and the Y chromosome has SRY, which are antagonistic against each other, causing only one to be expressed. A normal male has 1 X chromosome and 1 Y chromosome. The X is the DAX gene, and the Y has the SRY gene. Usually the SRY gene masks the DAX gene, but if a male is born with 2 copies of the DAX gene on the X chromosome, then he will genetically be male, but will phenotypically be female because the 2 DAX genes will over-trump the SRY gene that is on the Y chromosome. Because the X chromosome is present 3 times more often than the Y chromosome, the X chromosome is antagonistic and is always trying to shut down the Y chromosome. This is why the Y chromosome always tries to hide by shutting down some of its coding regions and leaving the really important specific regions, such as the SRY gene. The male in flies and humans has been shown to have something called seminal fluid. It is passed on in ejaculation, which is shown in fruit flies to pass on hormones and proteins that make the female ovulate more. The proteins also cause the female to want to mate with the male that gave her the proteins, letting the male have multiple chances at producing more offspring and passing on his genes. Females in return develop ways to resist such advances in chemicals. William Rice did an experiment where he mated males with resistant females and kept other females with a little resistance separate. He then mated the males with the females with little resistance, and the male's seminal fluid was so potent and had a super evolved protein that it killed the females.
The genome is, in essence, immortal. You are connected genetically to the very first thing that inhabited this earth, but humans, unfortunately, are not. That's because with each cell division and replication of our DNA, repeated sequences called telomeres, which consist of TTAGGG, are shortened. Now, the TTAGGG sequence is conserved across all mammal and many protozoans. However, the length, or number of times it's repeated, varies in humans from about 7 to 10 kilobase pairs. Your telomeres shorten over time as the cells replicate, so by the time you're 80 years old, they'll be approximately 5/8 the length they were at birth. Obviously, this would cause problems if the telomeres were to shorten in the germ cells. However, germ cells are protected by telomerase, an enzyme in part produced by the TEP1 gene on chromosome 14. Telomerase has important implications in aging. For this reason, you can find supplements that supposedly activate your telomerase for up to $200 for only 30 capsules on Amazon. However, telomerase is certainly successful at lengthening lifespan. In one experiment, cells were able to live for 20 more doublings when they were introduced to a telomerase gene that they were naturally lacking. An example of this in humans is the fact that arterial cells age a lot faster than vein cells do because they're under more pressure. For this reason, arterial cells tend to have shorter telomeres than their vein counterparts. One question scientists have puzzled over is why some animals seem to age more slowly than others, for example, elephants or birds. They realized this has to do with the fact that natural selection cannot act on genes that affect an organism after it's done reproducing. There's no selective pressure on these genes. Finally, the author discusses the famous HeLa cells derived from Henrietta Lacks. I found it interesting that if antisense mRNA is added to these cells to block their telomerase mRNA, they'll become normal and die after an average lifespan. These cells have tremendous applications in the treatment of cancer and could circle the globe 3 times if lined up end to end. They also weigh 400 times as much as Henrietta Lacks herself ever did. Chapter 15 is titled Sex and begins with a discussion of 2 different diseases. Prader-Willi syndrome results in children who are obese and bad-tempered, whereas Angelman syndrome results in those that are happy and strangely skinny. Interestingly, both of these diseases are caused by a missing portion of chromosome 15. Which disease depends on whether the afflicted chromosome was inherited from the mother or father. This difference is known as imprinting. To study imprinting, scientists created mice that had genetically only a single parent. They took early mouse embryos, which still had 2 pronuclei, one from the egg and one from the sperm, and they swapped an egg nucleus for that of a sperm. The result was one mouse that genetically had only a mother, which was well organized but lacked a placenta, and one that genetically had only a father, which was poorly organized, basically just a blob, but had a well-developed placenta. From this, the scientists theorized that the paternal genes are responsible for building the placenta, which can be seen as a parasite of the mother's resources. In a type of sexual antagonism, the mother's genes must battle for their fair share of resources, whereas the father's genes, whose interest lies only with their offspring, try to extract as much as possible as they can from the mother's body. As another example of this genetic conflict, scientists looked at 2 types of mice. One is promiscuous, so in the developing offspring, the paternal genes are used to fighting for as much resources as possible, whereas the maternal genes have a conflict of interest between the mother's body and that of the developing offspring. On the other hand, no such conflict exists in monogamous mice. When a promiscuous mother and monogamous father were mated, the result were mice of abnormal size. The mice were extremely small because the maternal genes had quote-unquote fought back and the paternal genes had done nothing. On the other hand, mating a monogamous mother with a promiscuous father produced extremely large mice. The paternal genes had fought to extract lots of resources from the mother's body, and the maternal genes could not respond. Finally, this chapter discusses the idea that genes for mothering behavior are present on the paternal X chromosome of females. If they were present on the maternal X chromosome, the book argues, these behaviors would be present in males as well. Evidence comes from a study of girls with Turner's syndrome, in which part of their X chromosome is missing. If the affected chromosome is maternal, the girls tended to be better socially adjusted. However, if the missing chromosome is paternal, they had greater social issues. The importance of this is that it explains that there may be a genetic difference, not just a socially imposed one, between the behavior of males and females. It also explains why males have a higher rate of conditions like autism that impair social function, because they inherit only one X chromosome with these social adjustment genes, and it is maternal, not paternal. Chapter 19, Prevention, focuses on apolipoprotein E, or APOE, a gene that's unusual because it's polymorphic, and its three forms, E2, E3, and E4, are all about equally common. APOE is important because it's implicated in worsening the effects of both Alzheimer's disease and heart disease, because it's involved in cholesterol and the formation of plaques, both in the brain and in the arteries. Despite these negative effects, scientists believe E4 must have some benefits, otherwise it would have been weeded out by natural selection. However, in families with high rates of Alzheimer's, someone with 2 copies of the E4 allele has a 91% chance of getting the disease, whereas someone with 0 copies has only about a 1/5 chance. Our ability to test people for which allele of APOE they have brings up a lot of new moral questions. For example, those with E2 can reduce their risk of heart disease dramatically by eating a diet lower in cholesterol. Don't we have a moral duty to inform them of this? What about people like boxers or football players, who are already at a higher risk of Alzheimer's because of their head injuries? Do we have a duty to tell them? Finally, there are economic questions involved. What about employers or life insurance companies? The book's author, Ridley, ultimately argues that your genome belongs to you. Unlike factors such as drinking or smoking, which are partially under the control of the individual, our genome is completely out of our hands, and so it should not be used as a point of discrimination. The book concludes with 3 major points. First, genes don't exist to cause diseases, in the same way your lungs don't exist to cause pneumonia. Second, genetics is a lot more complicated than we originally thought. Even though individual genes may act like binary on or off switches, when combined into a system, they become extremely complex. And lastly, we are only at the tip of the genetic iceberg. The future possibilities are endless.